cephalosporins, the citrusin, vancomycin, they do inhibition of cell wall synthesis. However, if you have a mycobacterium, you need to take an anti-mycobacterial drug. Because where is the mycolic acid? Where's mycolic acid found? In the cell wall. So here you are inhibiting the cell wall synthesis, but you have to take an anti-mycobacterial. To inhibit protein synthesis, know the mechanisms. You can bind to the subunit. You can stop the peptide bonds from forming. You can change the shape of the subunit. Remember, change the shape, change the function. You can stop tRNA from attaching. If tRNA and mRNA don't come together, you won't have amino acids coming together. You can stop translation altogether. You can stop the shifting of the mRNA. You can stop the ribosome from forming. There's all ways to stop protein synthesis. To stop injure the plasma membrane means to make holes or to actually fragment the plasma membrane. Once that happens, the cell contents leak out. Once that happens, the DNA, RNA, and proteins are left vulnerable in the cell. To stop the nucleic acids from assembling, either stop the RNA from being made. If you stop RNA from being made, you stop proteins from being made. Stop proteins from being made, then nothing in the cell is going to be expressed. You can stop DNA gyrase. So if your DNA can't be wound and unwound, then you can't replicate it. Competitive inhibition. You have the actual substrate, and then you have something that looks like a substrate. It's called an analog. So if the analog sits in the active site, then that's going to interfere with whatever the function was supposed to be. To stop your fungi, you inhibit ergosterols. The plasma membrane is pretty fluid. Sterols make it rigid. Take away the sterol, takes away the rigidity, takes away the integrity of the plasma membrane. Or, if you're not going to stop the sterols, then stop the production of glucan. Glucan and mannan are sugars found in fungi. So if that component's gone, then you would injure the cell wall. You can stop the nucleic acids, again, use an analog or bind DNA. You can stop microtubule formation. If you stop microtubule formation, what else can stop? Where do you see microtubules? Mitosis. So if you don't have microtubules, how are you going to undergo mitosis or meiosis? What else? Where else do you see microtubules? In the cytoskeleton. Now, the cytoskeleton serves as like a highway for things to move through. Now, if that portion is not made properly, then how are things going to move throughout the cell? Here's another example of inhibition. Kind of like competitive inhibition. So this is the important part. Hopefully you can see it. When you have a false nucleotide, it'll block DNA synthesis. So the drug serves as a false nucleotide. See, this is your nucleotide. This is the false one. This is the analog. This is the only part that really matters, the guanine, but once the rest of it is changed, the whole function would be changed. You could have enzyme inhibitors, protease, and integrase for HIV. So if you can't integrate, remember, HIV comes in the form of RNA as a nucleic acid, right? And an RNA would have you change the DNA, and then that DNA will be integrated into the host DNA. Well, if you can't, if you essentially disrupt the proteins that enable it to happen, then you won't be infected. Let's see, block entry, block fusion, or you can get injected with interferon. This is actually, alferon is an interferon. So get injected with the interferons or stimulate the production of more interferons. As far as the protozoans go, so if you're at risk for malaria, you can inhibit the DNA synthesis. You can kill the sporozoites of Plasmodium vivax, or you can interfere with the metabolism of these protozoans, so they can't break down anything that they take in. They then can't build anything that they need to grow. Stop ATP generation, alter the membrane permeability, or paralyze the worms. These are all ways to get rid of helminths.
So you don't have to know those fancy names?
No, just know the mechanisms. Do you know the difference between inhibitory and bactericidal?
Yes.
Yeah? You understand this is a ruler, right? Okay. Don't worry about this one.
Don't worry about it.
Yeah, no, please worry about this one. Worry about the antibiotic resistance. What can lead to antibiotic resistance? And let's say you take an antibiotic. It may not work, one, because the antibiotic does not get into the cell. If the antibiotic does get into the cell, the target molecule, see, that shape doesn't fit, like lock and key. You can inactivate the antibiotic. So remember, change the structure, you change the function, or the antibiotic stays intact but just gets spat out, essentially, by the bacteria. Do you understand what's going on here? No, you don't. E. coli has this plasmid that has this resistance factor, right? So what they did, they ran a sample of E. coli. They found the plasmid with the resistance factor to a certain antibiotic. But the turkeys didn't have E. coli. The infected turkeys had salmonella. When they ran salmonella, they didn't see that plasmid. So what they concluded was salmonella and E. coli conjugated, and so the salmonella afterward would then have the plasmid. Is this HFR or F plus? F plus. F plus gives the plasmid. HFR gives the bacterial chromosome. So that's all they do. They run the samples. Here's the plasmid in E. coli. It's not in salmonella, but if you mated these two, then salmonella gets the plasmid. Synergistic, you all did pretty good with that on the practical. Synergistic is when you have two zones of inhibition coming together. Additive, you understand those were apart, but they still worked. But see, antagonistic is if you got two drugs and they worked against each other. All right. Gene silencing. Here's your DNA. Your DNA has genes, okay? Make the mRNA. So you have your gene, your DNA. When you make mRNA, you're attempting to express that gene. Remove the introns, keep the exons, splice them together. But the thing is, this gene that was read actually is abnormal. So what you can do, there are these small RNAs that are, it's pretty new. The siRNA will bind to the mRNA and break it apart. So let's say you did transcribe a bad gene, then what if we can just inject you with siRNA and that will then target those genes? Or there's phage therapy where, again, you have the bacteriophage, the complex looking virus. Remember, it looked kind of like this. That was the head, and then it had the sheath, and then the tail pins. Okay, what if you actually put the drug in here that you wanted? And then when this binds to a microbe, it injects this drug into the microbe and kills it. Then this phage is essentially just a vector, right? Yeah, well, what you would have to do, you'd have to remove the nucleic acids from in the head, and then actually put the drug in there. Or what we're seeing is there are these proteins that we make, that animals make, that have antimicrobial properties. So what if you can then make that, like make more of that, and then use that in people? Didn't someone just tell me that they sell shark supplements?
Yeah.
So, maybe that's one of them. And this one, the frogs, they're looking at frogs that have spots on them, and that's what they're taking this from. That's it.